The plasma membrane is often considered to be the boundary of the cell. However, as we discussed last time, it is better thought of as a regulatory interface between what is inside the cell and what is outside the cell. Some substances, mainly nonpolar, can pass freely through the membrane. Other substances require help from a variety of different transporter proteins. This is referred to as selective permeability. The membrane and the proteins in it are capable of regulating what comes in and goes out. First, we need to look at the basic structure of the plasma membrane and examine how that structure imparts function. The basic building blocks of the plasma membrane are phospholipids. We saw them a bit in Chapter 3. Phospholipids are made of two hydrocarbon chains and a phosphohead group that are all covalently bonded to a glycerol. This is like a triglyceride with one of the hydrocarbon chains replaced by the phosphate head group. From what we have learned, we can extrapolate a lot about the properties of a phospholipid. First, we know that the two hydrocarbon tails will be hydrophobic, and that double bonds will make them pack less tightly. Next, we see that the phosphate group has lots of oxygens and can participate in hydrogen bonding. What is important about hydrogen bonding? That's right, it makes the head group hydrophilic. Remember tertiary protein structure? One of the things that stabilized it was hydrophobic interactions. This occurs when the water-loving parts of the protein face outward towards the water, and the hydrophobic portions are protected from the water inside. This same concept is how the membranes of your cells are formed. Again, structure implies function. The hydrophobic tail portions group up, and the hydrophilic phosphate head groups all face the water outside. This creates a lipid bilayer. The hydrophobic portions inside the double layer are protected from the water outside by the head groups. Let's think a little bit more about what we've already learned. We know that the double bonds make hydrocarbons pack less tightly, like we saw in butter and olive oil. Now we can apply that knowledge of a structure and function relationship to the membrane. The more double bonds there are in the tails, the less tightly the membrane will pack together. This makes it more fluid, like olive oil. However, lots of saturated tails will make the membrane rigid and less fluid-like. These lipids are free to move around in the membrane like this. This is called the fluid mosaic model of the membrane. However, other molecules are also found in the membrane and they help control the fluidity as well. Cholesterol acts much like a fluidity buffer to keep the temperature from having too great an effect. Proteins are also lodged in the membrane. Membrane proteins come in two flavors: integral, which means they're physically passed through the membrane, and peripheral, which means they stick to the outside of the membrane but do not pass through it. I bet we can guess some things about the structure of these proteins. Integral proteins are inserted into the membrane by the ribosomes stuck to the walls of the rough endoplasmic reticulum. The structure of these proteins is hydrophobic in just the right spots to sit happily in the lipid layer. Frequently, these are alpha helices with lots of hydrophobic side chains. Peripheral proteins, on the other hand, have most of their hydrophobic parts tucked away, and I bet you can guess what is on the surface. That is right, the hydrophilic amino acids that can bond with the phosphate head groups. Membrane proteins fulfill a variety of functions. These range from passive transport of molecules to the complex production of ATP. If we think again of the membrane as an interface, then the proteins are the controls. The transportation of particular molecules in particular ways is fundamental to life. So, let's take a look at the different types of proteins and mechanisms that are involved in regulating the flow of molecules across the plasma membrane. There are two basic types of transport proteins in the membrane. Channel proteins, which are basically just hydrophilic alleyways through the membrane, and carrier proteins, which can bind to molecules on one side of the membrane and pass them to the other. The most simple transport proteins facilitate passive diffusion. When you open a perfume bottle in a room, the scent slowly fills the space. That is because there is way more scent in the bottle than in the room. The perfume molecules diffuse passively out of the bottle, looking for an equilibrium, whereas the same concentration of perfume inside the bottle as outside. Molecules work the same way across a membrane. If the outside of the membrane has a lot of sodium, and the inside of the cell has very little, passive diffusion will start to fill the cell up with sodium from the outside until the concentrations are the same. This is an equilibrium, because once the concentrations are equal, sodium will pass in and out of the cell at equal rate. None of this requires any energy to be used. It will just happen, given a difference in concentration between the two spaces. Sodium will likely pass through an ion channel. Ion channels allow particular ions to pass through them by diffusion, or another way of saying that is that they are falling down their concentration gradient. An important example of passive transporters are aquaporins. These are channels that allow water to passively diffuse into and out of the cell. Water will flow in the direction that equalizes the concentrations of all the molecules inside and outside the cell. If there are more sodium ions outside the cell than inside, and they are blocked, the water will move out of the cell to balance their concentration. The opposite is also true. This process is called osmosis, and we refer to the number of molecules in the solution as a tonicity. Let's think about this for a second. Water seeks to equalize the concentrations of molecules, so it will flow towards the higher number of molecules to dilute them down. Scientists crudely say that water chases salt. So if there is more salt outside the cell, what we call a hypertonic solution, the water will rush out of the cell to balance it. However, if there is much less salt outside the cell than inside, a hypotonic solution, the water will rush in to balance it. If the concentrations are the same, this is called an isotonic solution. The danger for cells of hypertonic solutions is that the water can rush out of the cell and it will shrivel and lose function. What do you think the danger of a hypotonic solution is? That's right, the water will rush into the cell, causing it to potentially explode. The second class of transport proteins we're going to look at are also passive facilitators of diffusion, but these are carrier proteins. Passive means that they don't use any energy, but these proteins are not just open channels like ion channels or aquaporins. These proteins help accelerate the flow of molecules down their concentration gradient. That is, from an area with high concentration to an area with low concentration. Are you tired of all this passivity? Let's use some energy. Proteins that use energy to transport molecules up their concentration gradient are called active transporters. Where passive diffusion, like perfume from a bottle, will always go from a high to a low concentration, or down its concentration gradient, active transport will use energy, often from ATP, to push molecules up their concentration gradient. The equivalent of vacuuming all the perfume from the room and putting it back in the bottle. That seems like a lot of work, huh? Well, it is, and ATP is a powerful source of energy for the cell. A good example of this is the co-transporter lovingly referred to as the sodium-potassium pump. This protein is a co-transporter because it moves two different molecules together. In this case, sodium and potassium. These molecules are pushed up their concentration gradient. This transporter allows for high concentrations of sodium outside the cell, and high concentrations of potassium inside the cell. Why would the cell spend so much energy on this work? It seems kind of insane. Well, the reason that cells do this is so that they can use this potential energy to do work later on. But because sodium is a cation, there is an electrical gradient. Sodium is positive, and the cell is filled with chloride, which is negative. So if you open sodium channels, the chloride's negative charge will pull the sodium into the cell even faster. This is the basic mechanism that allows nerves to fire. The passive channels open, and the sodium rushes in. Pow! The nerve sends a signal. Then our friend the sodium-potassium pump works quickly to reset the nerve so that it can fire again. Otherwise, nerves would run down and stop working. Now, that's no fun. We talked a bit last time about how vesicles from the Golgi apparatus can fuse with the plasma membrane and release their contents. We've also seen food vacuoles, which take up things from the outside in much the opposite way. These processes are called exocytosis and endocytosis. Exo means far away, and endo means close by. And then cytosis means related to cells. Thus, endocytosis means bringing things into the cell, and exocytosis means pushing things away from the cell. The plasma membrane is a fantastic regulatory interface between the inside and the outside of the cell. Now that we've looked at how to physically move molecules from one side of the membrane to the other, in a variety of ways, in the next video, we'll take a brief look at how external signals are transmitted across the membrane and can affect cell activity.